we will go to the first speaker from the regulatory parts, and it will be Katherine Tyner from FDA. I will give you the floor, and we are listening to your talk.
All right. Thank you, and good morning. Thank you for inviting me to speak today. All right, we'll put up the FDA disclaimer really quickly, and then move on to the actual topic, which are complex drug products. So a couple of general points from FDA. So there's no definition from FDA of nanotechnology or related terms. So that includes nanomedicine, nanodrug, nanopharmaceutical, and also non-biological complex drugs. What we do have are the terms drug products containing nanomaterials. And since I am speaking under the disclaimer, I can say that for someone that speaks as quickly as me, nanomedicine is much easier to say. Then we also, instead of non-biological complex drugs, we have complex products. And I'll go into a couple of slides about how FDA looks at those. The other thing I want to point out is that regulations and laws do not separate these products. So they go through the same regulatory pathways, and they're not categorically judged as either intrinsically benign or harmful. So when you look at the breakdown of nanomaterials that FDA has seen coming into submissions, one thing you'll see is we have a huge diversity in the types of materials that we see. But you will see some of the bigger slices of the pie are liposomes, the iron colloidal products, and emulsions, which were brought up earlier and are oftentimes considered to be very complex. So FDA does discuss complex products. And in terms of the complexity of how we look at these products, they fall into different classes. So we have complex active ingredients, and those are your peptides, your complex mixtures, and naturally sourced products. We have formulations, so liposomes, emulsions. And when we talk about nanoproducts, a lot of those will fall into those first two, but also complex routes of delivery, things on the skin, creams. Dosage forms, transdermal systems are a great example, drug-device combinations, and then our favorite catch-all, all other products where there could be complexity or uncertainty. So that allows us to have a catch in case something comes out of left field that we had no idea was going to hit us, but also takes care of things such as like abuse deterrent formulations. Now, as mentioned, there was a draft guidance that was published at the end of last year. It's between the Center for Drugs and the Center for Biologics. Two main points that I want to talk about for this guidance. The first are that to reiterate that these products are regulated the same as products not containing nanomaterials. So they're looking for the same regulatory standards for safety, efficacy, and quality. The second is a risk-based approach, and looking at a risk-based approach not just for the development of these products, but as well as the assessment of these products. And the one part that I think we're all going to agree on is how key the nanomaterial. There we go, early in the morning. Nanomaterial attributes relate to the product quality and safety and efficacy. Since we're on the topic of guidance, we do have other guidances that fall under for nanomaterials. We have the overarching agency guidance, we have the draft guidance, and then we also have some class-specific guidances that can help for complex products, including the liposome drug product guidance, which we're going to talk about in a little more detail. And then we also have product-specific guidances, and the website here to take you to the list of the product-specific guidances. We have thousands of them. They're based on the active ingredient, and some of them do include what would fall into complex drug products. The product-specific guidances are specifically for the development of generic drug products. They are used in the Office of Generic Drugs and focus on determining bioequivalence. All right, so let's talk a little bit about the liposome drug product guidance. So this was finalized last year, so 2017 was a big year for nano products. And unlike a lot of the other terms, liposomes were actually defined within the guidance. I'm not going to go into the definition. I think the people here have a good idea of what liposomes are. And what they are is very complex. There's a lot of complexity in different portions of the liposome drug product, including the liposome itself and how that liposome is formed. So even the structure of the lipids can impact the function and stability of not only the liposome, but then the resultant drug product. They can also impact the safety. So if you have a double-tailed lipid, when it becomes single-tailed due to chemical degradation, it can form lysolipids. That's bad. So when lipid form, they can bind to red blood cell membranes and cause hemolysis and perturb the cell membrane integrity. Because of this, the raw materials, so the inactive ingredient of liposomes, are considered critical raw materials for liposome drug product manufacturing, and they include a lot of things such as a complete degradation profile to accurately determine the potential safety risks and the quality risks for this product. Lipids are obviously important for liposomes, but there are other inactive ingredients that also impact the quality, safety, and efficacy of these products, including things such as cholesterol. So cholesterol can be embedded within the bilayers. It helps promote the fluidity of the bilayer, reducing or increasing drug leakage. You can have PEG that is put on the surface to promote longer circulation times. And so a lot of times the concentration of those inactive ingredients and the ratio between the lipids and the other inactive ingredients tend to be critical factors that are going to determine the quality, safety, and efficacy. So when you look at those critical quality attributes, that's one of the ones that's specifically called out as something that should be taken a look at, because it can impact not just the quality, but also things such as the performance, including the PK/PD of that drug product. And then my very, very favorite part of complexity, and this is the physicochemical testing. So I'm a materials chemist by training, so I love playing with analytical techniques, especially the new ones. So when we talk about the physicochemical testing, these are complex products. A lot of times the analytical techniques applied to them are also complex. So suitable analytical methods need to be employed to properly characterize these drug products. However, like I said, the products are complex. The analytical techniques can also be complex. And if you use inappropriate methods, or if you use appropriate methods inappropriately, you can produce false results, thereby calling into question the data reliability and the product quality. Particle size is considered a critical quality attribute, and this is in the guidance. And it's something that we have across the board for liposome drug products. That's because the size impacts so many different portions of the performance of this product, including ADME, stability, and drug release. Because it's such a critical quality attribute, there's oftentimes requested that there's more than one way to measure the size. That includes dynamic light scattering, electron microscopy, and you'll see this for the critical quality attributes that impact so many different areas of the product. More than one technique can be recommended, especially when we're talking about something that's complex, to help get a handle on those critical quality attributes. Okay, so there was a great discussion yesterday about size and how important it is or is not. Size for liposomes, it's important, but it's not the only thing that's important. So for liposomes, they're used to deliver drug products. Looking at in vitro test of how that product is released from the liposomes also is something that is critical to help determine the quality of these products. So they're typically used to evaluate product quality and suitability of in-process controls during manufacturing. The in vitro release method should be able to discriminate between acceptable and non-acceptable batches of the drug product. Excellent. So size matters, in vitro release matters, and there's a whole host of other things that can or cannot also matter depending on how that liposome drug product is actually put together. So things such as morphology, surface characteristics, the liposome structure integrity, and the leakage rates. Some of these are hard to characterize and hard to get a handle on. And so when we start talking about those complex physical chemical testing, this is where we do a lot of the work and a lot of the research that is done at FDA. So my group put together an analysis. We took all of the INDs, the NDAs, and the ANDAs, all of the applications for liposomes that have come into the FDA over the years. And we did an evaluation of categorizing all of the questions that we had between the regulators and the companies. So any of that back and forth was documented and captured. It was bucketed And then we did analysis to see where those questions were. Where were the problems for development? Where were the questions for assessment? And the two main challenges that were identified for both development as well as assessment were the identification and appropriate characterization of critical quality attributes and how to characterize them. And then related to that, what are the suitable control strategies during manufacturing to be able to keep those critical quality attributes within specifications? So I mentioned a bit about product-specific guidances. These are for generic follow-ons, and this is the one that we have for liposome drug products. This is for doxorubicin hydrochloride liposome injection. And so a bit about generics. So as I mentioned, FDA regulates these the same way, all these complex products, the same way as we would products that are not considered complex. They go through the same pathway. So like all generic drug products containing nanomaterials, they're going to meet the following general criteria, and this is that they're approved, safe and effective, they're pharmaceutically equivalent, which I think we'll have some good discussion about, they're bioequivalent, they're adequately labeled, and they're manufactured in compliance with CGMP regulations. It is the ANDA applicant's responsibility to demonstrate that you have equivalence between the generic and the reference material. It's our responsibility at the FDA to assess that and make sure we agree with that. The key is the comprehensive characterization of the RLD and understanding of the fundamental chemistry used to form that active ingredient to demonstrate equivalence. When you have a complex drug substance, that is a complex question. If we talk about parental products, where a lot of these non-biological complex products fall into, there are some additional challenges that should be considered for looking at physicochemical sameness. The active ingredients for some nanomaterials are heterogeneous mixtures. There's critical excipients that can impact the quality and safety of these products. The properties can depend on the manufacturing process, can be very sensitive to that. And the drug levels in systemic circulation may not always be reflective of what the actual drug concentration is at the target. Evidence of comparable PK parameters in blood and plasma in conventional BE studies alone may not be sufficient to satisfy the requirements for approval, and so that's where we get back into the physicochemical testing, looking at different ways to help ensure that you have similar ingredients, and that includes particle size, surface property, the in vitro drug release test, and then things such as free and nanomaterial-associated drugs. So, very complex, and I have 30 seconds to go over that. There is help available, at least discussions with the FDA. So, starting in October, there was established what's called the pre-ANDA program. Its whole goal is to clarify expectations and ultimately promote a more effective and efficient review process for generic drug products. It includes 3 meetings: the product development meeting, the pre-submission meeting, and a meeting after the ANDA applicant has been started to be assessed. So this is all face-to-face meetings with the agency to discuss these complex issues. And I see that my time is out, so let me just, I have a reference slide of how you submit a meeting request to actually talk with us. And in summary, we see a large portfolio of diverse and complex materials. The complexity and the diversity is increasing. Characterization is key. There's guidance available to assist, as well as discussions with the FDA to assist in the development of these products. And with that, I'm happy to answer any questions.
Thank you very much in not only keeping the time, but giving quite a nice overview on what is the focus of FDA